Hello everyone, you are welcome to my YouTube channel. I'll be discussing antimicrobial susceptibility testing. Antimicrobial susceptibility testing is usually done to aid the clinician in the selection of antimicrobial agents in the treatment of infectious diseases. Specifically, I'll be discussing the Baeur NCCLS modified diffusion technique. And this involves preparing turbidity standard equivalent to McFarland 0.5. Briefly, the turbidity standard is actually a barium sulfate standard against which the turbidity of the test and control inocular can be compared. First, 1% volume by volume sulfuric acid is prepared by transferring 1 ml of concentrated sulfuric acid into 99 ml of distilled water. This will be followed closely by preparation of 1% weight by volume of barium chloride solution by weighing 0.5 gram of barium chloride and dissolving in 50 ml of distilled water. In order to prepare the barium sulfate standard, 0.6 ml of the barium chloride solution is transferred into 99.4 ml of the 1% volume by volume sulfuric acid. Also, control strains from reference laboratories should be obtained to test the performance of the method. In order to prepare the bacterial suspension of the test organism, as well as that of the control inoculum, a sterile wire loop should be used to touch 3 to 5 well-isolated colonies of similar appearance to the test organism or control organism, and then emulsified in 3 to 4 ml of sterile physiological saline or nutrient broth. Now, the turbidity of this bacterial suspension should be matched in a good light to this turbidity standard. That is the barium sulfate standard. Here is an example of McFarland standard prepared, specifically McFarland 0.5, which is used in this technique. Also, a plate of molar Hinton agar will be required to inoculate the test organism, and another plate will be required to inoculate the control organism using a sterile swab. Excess fluid should be removed by pressing and rotating the swab against the side of the tube above the level of the suspension, as shown in this figure here, by pressing or applying pressure to remove the excess liquids from the swab before inoculating the molar Hinton agar surface. The surface of the medium should be streaked evenly in 3 directions to ensure confluent growth. Rotating the plate approximately 60 degrees to ensure even distribution. With the Petri dish lid in place, 3 to 5 minutes are usually allowed for the surface of the agar to dry. 10 commercially prepared paper antibiotic discs or antimicrobial discs with known disc potency are placed on the inoculated agar surface using sterile forceps needle mounted in a holder or a multi-disc dispenser evenly distributed on the inoculated plates. Plates are incubated at 35 degrees Celsius for 16 to 24 hours. The zones of growth inhibition around each of the antibiotic disc or antimicrobial disc are measured to the nearest millimeter. The diameter of the zone is related to the susceptibility of the test organism and to the diffusion rate of the drug through the agar medium. The zone diameters of each drug are interpreted using the criteria published by the Clinical and Laboratory Standard Institute. Here is a figure showing the plate, molar hinting agar plate with the disc and the zones of inhibition. And this is usually measured. The zone of inhibition is measured using a metric rule or ruler across the zone of inhibition. Using the interpretative chart of the CLIS, the zone sizes of each antimicrobial agent is interpreted, reporting the organism as resistant, intermediate, or moderately susceptible, or susceptible. There are selected references for further reading. Thank you for listening.